the difference between sensation and perception? Because we know there is a difference. Yes, they work hand in hand. Can we perceive something in the absence of sensory input? That's true. That's called hallucination. Sensation is any information we bring in through our senses. Hearing, smell, vision, taste, touch. Our brains only know how to process neural impulses. So right now we're going to talk about vision. So the visual input that we are taking in through our eyes, at some point, the colors, the shapes, everything that we are seeing at some point needs to be converted into a neural impulse. That conversion is called transduction. We have this information that is flooding us, coming at us. And at some point, that visual input has to get all the way to our occipital lobe in order to be properly processed so we see what's out there. So light is going to enter in through our eye, and we're going to break down the anatomy here. Okay, so it's a really small amount of stimulus necessary. First come in contact with our cornea. If you are a contact lens wearer, the cornea is where your contact sits. Cornea has a job to bend the light inward. It's going to come in contact with this gap right here, and this is the anterior chamber filled with fluid, and that fluid is called the aqueous humor. It gives the cornea nutrients. Now we're going to come in contact with two structures that are kind of on the same plane. That would be the pupil and the iris. Our pupil looks like a black circle in our eye, but actually it is an opening where the light goes through. The iris is actually a muscle. It dilates, which means gets bigger, and contracts, gets smaller, depending on the amount of light in the room. Interesting thing about the lens, it's got an important job in that it focuses the light onto the retina. The lens is a little bit flexible, so the muscles will adjust the shape ever so slightly of the lens, which allows us to focus. And so if this lens is very cloudy, then it's going to reduce the amount of light getting through, which is going to reduce the amount of information. Over time, our lens becomes less flexible. You may have heard of presbyopia. Old term for it is old eyes. When someone in their 40s or 50s has a hard time seeing, it's because the lens has lost a little bit of flexibility, not being able to see very well close up. Now the light is through the lens, and now you see this here, the vitreous chamber, which is filled with the vitreous humor, the fluid. The light is going to finally come in contact with the retina. When we talk about the retina, that is responsible for transduction. Looks like it's yellow. That is the retina. So it's a very, very thin sheet of cells at the back of the eye and inside the back of the eye. The retina contains really important receptors that do the transduction. In this case, of light to neural impulses. Yeah, the light would stop there. There would be nothing to make the conversion, and so no input would go to the brain. Let's talk about why the retina is so important and what kind of specialized cells they have. The retina contains photoreceptors. They are called rods and cones. Cones are specific to color, high visual acuity, how well and how detailed you can see. Your rods will help you with in any environment where the lighting is very, very low. You could see the outlines of people. You would kind of know where to walk and where not to walk. That would be your rods at work. Collectively, we call those two photoreceptors embedded within the retina. It is the photoreceptors that do the transducing. Terms that are for you to look up on your own: macula and fovea, central vision versus peripheral vision. So there we are. Now we've got neural impulses, information in neural impulse form. Now it's going to travel down the optic nerve. The optic nerves come from the back of each of the eyes, okay, and then they cross over at what we call the optic chiasm. The information is traveling on the optic tracts. Not quite to the occipital lobe yet because it's got to make a pit stop to the thalamus. Yep, it takes sensory information and directs it to the appropriate part of the brain for functioning. The thalamic nuclei have very specific areas where different sensory input. It's not enough just to say the thalamus. Specifically, you need to know it's the lateral geniculate nucleus of the thalamus that processes visual information. Okay? So after the retina transduces the light into neural impulses, then those messages, those neural impulses travel along the optic nerve. They cross the optic chiasm. They travel on the optic tracts to the lateral geniculate nucleus. That part of the thalamus goes to the occipital lobe. Specifically, it's the primary visual cortex of the occipital lobe. And that is how we see. Do I expect you to memorize this? Absolutely. Once it gets to this part of the brain, now it's perception. So sensation is taking in the sensory input. Perception is detecting, making sense of sensory input. So sensation, taking it in, perception, making sense of it. You guys remember what the superior colliculus or the superior colliculi do? Visual information that you didn't necessarily anticipate, it just kind of shows up in your visual field unexpectedly. Some of the information from the optic chiasm is processed in the superior colliculi. Optic nerve, optic chiasm, optic tracts, most of it goes to the thalamus on each side, a little bit of it goes to the superior colliculi. Allows us to orient to unexpected sudden visual stimuli. The striate cortex is an older term, now we call it the primary visual cortex of the occipital lobe. Primary meaning first. Okay, so first the information goes there. This information being processed by a part in our hypothalamus called the suprachiasmatic nucleus, and that sets our circadian rhythm. An overwhelming majority of the neural impulses go from the optic tracts right to the thalamus. Where did the other stuff go? Some of it went to the superior colliculi, some of it went to the suprachiasmatic nucleus. Our optic nerves can be smaller than they should be, but that doesn't necessarily cause blindness. A severing of an optic nerve, different story. A malformation to the point where there was no connection or the connection's in the wrong place, it's a problem. If someone has a brain injury and their occipital lobe is absolutely non-functioning, no vision. What if the thalamus is damaged and the thalamus sends that information to the temporal lobes by mistake? No vision. You see all the places where blindness could occur? If you know this process inside and out from exam day, you can answer any question I throw at you. So it turns out that the cells of the retina are more than just the photoreceptor. We have bipolar cells. You see this image, what we would call a horizontal cell. Another, only one, which we call an amacrine cell. And then we have the ganglion cells. Axons of the ganglion cells make up the optic nerve. Super important. I probably write that down. The rods and the cones have to do the transduction. This information, once transduced, has to actually go this way in order to travel along the optic nerves to get to the thalamus and occipital lobe. The light comes all the way down, is transduced here, and then the information is sent back up and then travels along the optic nerve. So this is just a little cross-section of the retina. We know that the light eventually has to get to the retina, but the way they have this in the ganglion cells up there that make up the optic nerve and the rods and cones down here is very confusing. The light does come all the way down. This is where transduction takes place, rods and cones, remember? From there, it eventually gets to the optic nerve and then can travel to the thalamus and then where it needs to be. We'll start with the ganglion cells. You already know the ganglion cells, but probably the most important thing that you need to know there is the axons form the optic nerve. The amacrine cells helps to process movement. We notice and we can understand that objects around us are moving. And that allows us to do things like walk and drive. And the amacrine cells integrate visual information from other cells. The bipolar cells help us to identify contrast, which allows us to see boundaries and edges. The horizontal cells, they integrate information from the photoreceptors. Here with what scotopic vision is and photopic vision. So primary visual cortex is the first part of the occipital lobe that processes vision. That allows you to detect that a visual stimulus was there. We need other parts of the occipital lobe to give us more information. From our primary visual cortex, that information gets processed by our extrastriate cortex, or our secondary visual cortex, allows us to know more information about that thing. What it is, where it is, the details of it. Right? So our dorsal stream is the pathway from our primary visual cortex toward the parietal lobe. That is called our where pathway. Parietal lobe is for processing touch, but our parietal lobes are also for understanding visual, spatial information. Hence the name where pathway. So our primary visual cortex allows us to detect visual stimuli. The where pathway tells us where the visual image is, also known as the dorsal stream. The PVC tells us something's there. The secondary visual cortex tells us where it is. It also tells us what it is. So let's talk about the ventral stream. The ventral stream, so the what pathway, is primary visual cortex to the temporal lobe, and that helps with object recognition. Tells us what it is. In our environment, the more important part is the activation of the secondary visual cortex. That tells us where things are. That tells us what they are. Being able to recognize what that object is going to help me know what to do next. This original detection, the primary visual cortex, that has to happen before the secondary stuff can happen. Are they going to show what we call functional impairment? Yes, they can see it, but they don't know what to do with any of that information because they can't make further sense of it. Yeah. Okay, do you think recognition of a person is important in our regular everyday life? In our temporal lobe, fusiform face area. So our medial temporal lobe is what we call our fusiform face area, FFA. Is a very specific function of the what pathway. There are people who absolutely 100% cannot recognize people by face, including themselves and loved ones. How else can we recognize them? And they actually function remarkably well. They just cannot identify people, even who are familiar to them, by face alone. Information on depth perception, you definitely want to read that. Okay, so depth perception, this is the idea that both eyes have to be present for three-dimensional depth perception to be a thing. Because each eye sees a little bit of something different, and when they bring that information together, that creates the perception of depth. You read up something called binocular disparity or retinal disparity. So, disorders of the visual system. When we see someone whose eye, one eye is tracking, the other eye is floating. When one eye starts to drift, or you can't really tell, looks like they're focusing on one eye and this other eye, you're not really sure where it's looking, that's called strabismus. Depth perception is off. You need both eyes focusing on the same thing in order to be more likely to fall down stairs or have problems with understanding how close or far something is. Patch the strong eye with the idea of getting this one strong on its own. Sometimes patching works. They're going to lengthen this muscle and tighten this muscle. Does that make sense? To bring the eye back online. Either patching or surgery will address strabismus. Amblyopia is when the information that's taken in through the drifting eye, your brain stops processing it entirely. We don't want it to start participating in other processing because then that person will lose processing in that eye entirely. So, strabismus is the drifting eye. We need to intervene, or this will lead to amblyopia, which is where the drifting eye, the information it brings in is no longer processed by the brain. You can't ever retrain that part of the brain to process vision again. Strabismus surgery in babies, like if they start to show strabismus, it needs to be addressed as soon as possible. Do you see the cloudiness in the lens there? Cataract is something that can happen to people as we age, but it can also happen based on, there are genetic conditions where someone is born with cataract in one eye or cataracts in both eyes, or they develop it very, very early on in life. You guys can read up on myopia and hyperopia. That's nearsightedness and farsightedness. I can't have the fancy words for that. Read up on astigmatism. This is permanent. It's areas of the brain, the occipital lobe, that have been damaged. And then the very last thing, the visual agnosia. So you guys see that on the very bottom of your outline. And this is when you can see the object or stimulus, but you have difficulty recognizing what it is. They can see it, so they've detected it, but they don't recognize it as familiar. Agnosia is a name for unknowing. So they can see it, they just can't recognize what it is and what it's for.
So what?
So we have to ask it in a particular way. We don't say, what is this? We say, what's this for?